Alright, here we are reviewing the PowerPoints for Chapter 5 and in general membranes and membrane structure, the little proteins here and there. But let's look at the membrane structure. Phospholipid bilayer, so that is something you need to know what that is. That has been mentioned in other chapters before. On the other tests, you need to understand what the how the membrane is put together. We're going to review it a little bit here, but not much. You need to understand hydrophobic and hydrophilic. You also need to know in this test for sure, or let's say as a central idea here that we need to understand is that within the membrane, it's not just lipid. You have proteins and you can have carbohydrates also embedded in the membrane. The membrane, of course, has two layers of lipids, so it is a bilayer, and there are all kinds of different things in the layers. Something can go all the way through, or it can only go partially through the membrane, but there will be proteins and carbohydrates that are stuck, and other molecules, some modified lipids, for instance, that will be stuck in that membrane and will give it some different functions, all right, that we need to learn about. All right, so like I said, lipid, protein, and carbohydrate molecules are all in there, and they can move around, all right? It's fluid. So here is our picture of that, and of course here you see the lipids. All right, now here's one with a kink in it, so that's cholesterol. If you ever see this, you know that's cholesterol, and cholesterol is there. A lot of times your body will put a cholesterol molecule in there because it does not, once temperatures and different things cause the temperature drop, and some other things can cause the membrane to become very rigid and could break. And when that occurs, if you put a few cholesterols in there, it gives it more flexibility so that it doesn't break. That's why you would want some cholesterol in there. But here is your, as you can see, they have a cholesterol here. Let's see, here we have a glycoprotein, which means it's a carbohydrate stuck to a protein. The little yellow piece here is the carbohydrate part, and here's a protein. Usually these things are not single proteins like this. They are a complex of proteins, a group of proteins that are all stuck together, and then those are stuck into the membrane. But you could have single proteins here, and all of these little colored blobs here are various proteins. So an integral membrane protein is one that goes all the way through, and here we have another protein that is stuck to the bottom of that. That could be used, actually they're saying, let's see, that's the cholesterol. I was thinking they were saying it was cholesterol, but no, it's another protein stuck to the bottom of it. And so realize that these things are not just one protein, there are often many proteins stuck together, and we call that a protein complex. All right, so we have transmembranes. Those are the ones that we're saying integral membrane proteins. Some are anchored to the lipids, as in they're bound. They have some type of bond that's attaching, covalent bond here that would do it. And others might be peripheral. They are bound but not covalently. Remember, the covalent bond is much stronger than the other type, like electrostatic or hydrogen bond that you might have, and it's really just lightly stuck there. But a covalently bonded protein would stick much more readily to the lipid bilayer. Now here we have the extracellular and cellular environment. We mentioned the matrix in a previous video, so that would be what's outside the cell. And here's the cytosol or the cytoplasm. This is internal to the cell. All right, so here is one that's anchored here, and here we have two proteins, one that goes all the way through and one that's bound to the outside here. And This here shows the structure of the protein. Proteins often have these structures here, either something called a beta sheet or an alpha helix, and they will be in a kind of assembled, and then they're just kind of attached by a loose turn here. And so this is a transmembrane alpha helix, and so this particular protein is made out of, it looks like 7 alpha helices, maybe even an 8th one that's sticking up outside of the protein. I mean, outside of the membrane here. And so an alpha helix is just a shape, a structure that the protein can form once it folds up. Let's see, 20 to 30% of all genes encode for transmembrane proteins. In other words, the majority of your genes are proteins that are stuck in the membrane, which is important, as it says here, medically as well as biologically. When these things don't work right, you have usually some type of illness. You can't get the things in and out of your cells at the right time or in the right amounts, and therefore you become sick. Your cells die or they don't function very well. And realize that the function of many genes are still unknown. We know something is a gene, we know that it makes an mRNA, that makes a protein, and we can't figure out what they do. So keep that in mind, that many genes are still unknown. We know quite a bit about humans and mammals, but for a lot of species, we know there are genes there and we have no idea what they do. I've looked at something, there's an algae called EHOX, and it's one of these really odd genomes. When they look at it, they say, oh, there are a bunch of genes here, has a lot of genes, but we can't figure out what most of them do. So it's a really interesting thing, and that's, people study those things to look for new functions, like to create drugs and things like that, and compounds that are unknown, or compounds that might be useful to us that we've never seen before. Nature gives us an example of how to do something that that we don't know how to make right now, but you'll look at some of these things that different organisms, plants especially, do, and we copy it chemically, and we essentially use it like inspiration for inventions and things like that at the chemical level anyhow. Alright, so fluidity just means that the membrane is not rigid. It means it can move around a little. You don't want it to be too rigid. So you want to keep it fluid. You don't want it to be too fluid or it'll just kind of fall apart. So membranes are semi-fluid. Let's see, lipids, not that important, but you do need to kind of know about this flipase. All right, and it requires ATP, so this is a powered function. It requires energy to do the flipping, but what the flipase does is that it takes something on one side of the membrane, and the protein, when something attaches to it, it flips around to the other side, kind of like a rotating door. If you've ever been in a large office building and they have one of those doors that you rotate through, that's kind of what the flipase does, and whatever gets trapped in it kind of rotates into the cell or out. All right, so keep in mind that's what a flipase is. It's kind of a way, it's a protein that when something attaches on the outside, it can flip whatever attached to it onto the inside. I guess maybe I can draw something like that for you. Let's see here. I got to get the right. This is a good one. All right, let's draw that. Okay, so here is your membrane here, and let's say there is a protein stuck in the membrane here, and we have some type of molecule here. Maybe another protein. It's a chemical signal from another cell that it's released. Well, there will be a piece on usually some type of what we call a receptor on this protein, a place that physically and chemically attracts and combined with this particular protein that's coming from somewhere else. And it will be attracted to this and it will bind. So let me draw it again here. And the little red piece will stick. Alright. And when it does, it causes chemical changes within the protein or protein complex that is here. So the shape changes is what I'm telling you. And ATP might also be involved. So there may need to be ATP here for this to occur. So that might be an additional chemical that's needed around here. And of course we rip a P off, I mentioned in one of the other videos, and we turn it into ADP plus inorganic phosphate, which we draw as PI. So in other words, ATP is adenosine triphosphate, tri meaning 3, adenosine diphosphate meaning 2. So 2 phosphates here, 3 phosphates here. We rip a phosphate off, and what that does is that adds energy to the proteins. And so now they have energy, and what will happen is that this orangey looking one will change its shape. So it changes its shape somehow when you add the energy. Alright, so you changed shape, alright, when you added some energy plus when this thing stuck, and what it does is it rotates. So think of it this way, when all of this occurs, the shape change occurs, and now you have it facing downward. I changed colors on you. Alright, so it flipped around. Okay, it rotated. It flipped. Alright, and now whatever this protein was that was on the outside, it has flipped around and taken it onto the inside. And then usually when it gets inside, the chemistry out here may be something like the pH or the salinity or something else is different from the inside to the outside. And what happens is the chemistry in here is such that now the little red protein here cannot stick any longer. So it was fine out here, flip around the other side, but on the other side the chemistry on the inside of the cell is different than the chemistry on the outside. And so the shape changes again, either the red one or the flipping one, or maybe both, but they change shape in such a way that now they can no longer stick. So now you end up with this protein here, and again I changed colors on you, and the little red one falls off and now can go and do whatever it needs to do inside the cell. So it's usually a chemical signal or something. So sorry about changing the colors. I should have tried to keep that the same. But the red one will stick here, causes a shape change in the little yellow one, and when that happens it causes a flip, and then once it's inside, now there's chemical difference between the inside and outside. They change shape, probably the red one, but it could also be the one that's in the membrane as well. They change shape, and now they can no longer stick to one another, and now this is free to go do whatever it wants to do inside the cell. Alright, hopefully that is somewhat clear. Alright, so let's take a look at this. So here they're showing this. This is what I'm kind of showing you. It flip-flops. And yes, there was some ATP needed. I drew it on the outside, but it could just as easily been on the inside. There would be ATP perhaps floating around on the outside of the cell too. If you're maybe not as common, depends on where you were, I guess, in the cell. But I guess this is more accurate, that you would have the ATP present on the inside to add the energy. But ultimately, you get this protein flipping from outside to inside. Let's see, certain lipids associate strongly with each other to form lipid rafts. Okay, lipid rafts are a more rigid structure. and you'll usually have some proteins associated with a raft. So it's kind of like the rest of the membrane is kind of more fluid, like water, than if you had a rigid structure there. But like I said, cholesterol is important here along with the membranes. Alright, so the length of the phospholipid tails. The length of the tails, they do affect the fluidity also. Double bonds, and of course the presence of cholesterol. So kind of in general know these three things: the length of the tail, the double bonds, and the cholesterol. Temperature also affects it. That's one reason why you might want more or less cholesterol. Membrane proteins are larger than lipids and can still rotate and move laterally. Realize that there's this a flip-flop is a transmembrane. Transmembrane goes from one side to the other. It actually spans the entire length of the membrane. And often these are not just a single protein, they are complexes. Oh, we don't have to worry about that experiment. I'm not going to do that, but you might want to know about this one. I might ask something here about a fusion experiment. So read this. I'm going to leave you to figure out what they're talking about here, but realize you can have mouse cells and human cells, and they can fuse and give them properties of both, a kind of a Frankenstein kind of a cell there, but that can be done. Now, not all of them move. You need to understand what the cytoskeleton is. So at least know a definition of the cytoskeleton and how it might be used to restrict proteins from moving laterally. And we've already talked about things that are outside the cell. We've talked about the extracellular matrix. Realize cells can attach to things that are in the matrix. And that's all it's saying here, is that just like proteins can be attached to carbohydrates, and you could have two cells attaching to one another. I drew a little picture of that in a different video. You might also be attaching to just something that's out there in the matrix, kind of binding you and holding you in place, more or less. The matrix is out there, and maybe you want to attach to some things that will kind of hold the cell in one particular place, make it more rigid. And here's the matrix, and see that it looks like fibers here, and this would be what you would attach this protein to. And then the cytoskeleton is also what gives the inner part of the cell its shape, and you would have it attached there. So you have this, and so this is a kind of a protein that kind of holds everything in place. It holds it from the outside and it holds it from the inside. So everything is kind of like really connected here at this particular protein. Here, this one isn't connected to the cytoskeleton, but it is connected to the extracellular matrix. And so, but this one here has both. It's connected to both. And so just to show you that such a thing exists. Now, the synthesis of lipids. In other words, where do they come from? How do you make them? Well, the whole idea here is that you have an endo, which is internal kind of, versus exo, system that synthesizes lipids. All right. Now you can get lipids from exterior. It can be brought in, but most of the time the cell is going to make its own lipids. And the process is carried out in the smooth endoplasmic reticulum, or we abbreviate that as the ER. And so this is basically your lipid factory, or a membrane factory. Actually, you make lipids and then you package them together into membrane. So you could call it a lipid factory, but you could also easily call it a membrane factory as well. I won't ask this directly, but you should read it. It's kind of the steps for how, what do you want to call it? How you kind of put everything together here. So I'll leave that for you to read, figure 5.6. Now the lipids in the ER, it's actually like pre-membrane. It's on the way to being part of a membrane of something else. They're made at the ER, and they just kind of float out to the outer part, and this just kind of merge with the outer membrane. There are vesicles. Now these are little like organelles don't have exactly a function normally, but they kind of transport things. So if something needs to get from one place in the cell to another, you might put them into a vesicle. All right. And lipid exchange proteins extract lipid from one membrane for the insertion into another. So in other words, maybe you want to take a few lipids, maybe the cholesterol. Maybe you want to take the cholesterol out of the outer membrane and put it into an organelle. Well, there are proteins that can do that. That's all it's saying here. That's a little bit detailed there. I probably won't ask a question on that. Let's see. You kind of need to know kind of the order of things. So the synthesis, I'm probably not going to ask you this much detail on the exam. You do need to know about vesicles, though. Vesicles kind of move. Oops, didn't mean to do that. Vesicles kind of move things around. They're like a truck in a city. If a cell was a city and you wanted to get something from one organelle, like one building to another building, you would package it up into a vesicle and send it over somewhere else. You would use a vesicle to do that, and so that way you can move proteins around to where you want them. All right, and here we show some proteins. We have a membrane here, and this is the membrane of the endoplasmic reticulum. And what they're doing here, they're creating membrane and they're stuffing it full of proteins. So how do the proteins get in there? That is done in the ER lumen. So this is an RNA here, and these are ribosomes, and they're actually making the proteins. They're actually translating them, and as they come out of the ribosome, they are kind of pushed into the membrane, and that's one of the functions of the ER as well. Is that not only can you make lipids and turn it into membrane, but you can also push the proteins you want in there into the membrane as the membrane is being created. A bit difficult. I might talk about this a little bit more at some point. I may not, but do realize that this is the ER is where you not only create the membrane, but you also put the proteins in the new membrane that you're making. Glycosylation is when you attach a carbohydrate to a protein or a lipid. So if you glycosylate something, you have added a carbohydrate to it. And usually carbohydrates are kind of sticky, so they're good for attracting things that come along and stick to the outside. So you put a certain shape on it and make it kind of sticky, and something that's shaped right will come along and stick there. And that's kind of the way we recognize what's on the outside of the cells. The cell knows what's out there, but depending on what sticks to it. And specifically sticks to these glycoproteins that are on the outside of the cell. I won't ask you the difference between the N length and the O length. Just know that the glycosylation exists. Here I've drawn something like this before for you, where you have this branched carbohydrate out here. So maybe watch that other video. And then there's transport. So realize membranes, one of their main function is to transport things. Now the membrane itself is selectively permeable. Some things can pass. Water can pass, but it's very slow. It's supposed to be waterproof, but it's really not. Water does pass through the membrane, just it doesn't pass very quickly through the membrane. It diffuses across the membrane is the way you technically should say it. Now if you want to get larger things across, you're going to have to use some energy and probably a pump, a protein pump that will pump things across the membrane, like that flip ace. The flip ace would be considered that kind of a pump or a pore. Ions, some very small things can pass without much problem through the membrane. Things like electrons and protons and things. They are they're pretty easy to pass through, at least in certain areas of the membrane. Now the structure, essential, how do I say this? Certain things you want in. So you're going to create pumps and pores that are going to bring those things that you want in. But you're also going to have things that are recognizing what's going on. So I've talked about that just a second ago. And those carbohydrates are going to be monitoring what's on the outside. Sometimes you want to create pumps and pores only after you know that something is out there. So there may be something out there that can signal that you need to create a pump or pore to let something in. So let me kind of draw that. Let's do a new one here. All right, we'll make it bigger. So let's say we have a cell. All right, and here we have, let's make it orange again. We have a protein stuck in the membrane, and it is particularly sticky. I'm going to draw it kind of like this, to kind of maybe it's a carbohydrate stuck on it. And basically we have, maybe that's exactly what we have, is something that's kind of like a carbohydrate. And notice I've made this so that it's shaped so that it can stick right in here. And so when it does stick to this thing, maybe it's a flip ace or something, but I'm gonna say it's just a signal. And so there might be on the other end of this another protein. And when this thing sticks, well, let's see here. When it sticks, basically it's going to change. The yellow one here is going to change shape. This little protein will change shape, and when it does, the red one cannot stick to it anymore and it gets released. So this thing sticks, all right, and when it does, this part changes shape, and that means this can no longer stay attached, and it falls off. Now this is a chemical signal, and this signal then can activate things. It can turn on and off genes. It might be able to get into the nucleus and say, hey, start making a whole lot more of something. And if it does that, maybe the thing it says to make is a pore. All right, so I'm going to draw, let's use a different color. And so it makes a couple of new proteins. And so these proteins I'm going to draw, and I'm going to erase the middle here. And it's a pore, and it's shaped so just the right shape, maybe, that these little things, whatever stuck here, can now get in. Oops. Why am I not drawing? Oh, I need to be here. Okay. So these are the right shape, all right, that they can fit in here, and you let them in. All right, so when these are around, they stick here. This is a signal to make these. So in other words, this one, when it sticks to the outside, sends a signal back to the rest of the cell that, hey, we need some of these proteins that can let this stuff in. And so you start making those, and sure enough, this stuff then can get in and out of the cell. and you might, maybe it's some kind of food or something else. But we do this is the kind of thing we do with insulin. All right, so I made an earlier video, or I used an older video, I believe, that I'd made a couple of semesters ago, and I described insulin. And so this is kind of where we're going with this. The insulin example is a very good one. I'll try to find some more videos on that. All right, so let's see. Let's do a couple more before I end this video. So let's see here. Passive transport is when you do not need energy, so no ATP is needed to get something across the membrane. So you're transporting something across the membrane, and if you don't need energy, it's called passive transport. If you do need energy, in other words ATP, then it's called active transport. And that's exactly what this is showing. Here is simple diffusion. Simple diffusion is probably going to be those ions, tiny things. Facilitated diffusion, all right, well, it's passive in that it's just got the right shape to let these through. You can think of it that way. Or there's a reason, maybe these are positively charged and the inside of the cell is negatively charged, and so these positively charged particles want to be in here. They're attracted to the negativity of the internal of the cell. Something like that. I'm kind of going out there without going into too much chemistry, but that might be it. But when you use ATP, all right, we call it active transport. And here we have ATP used to change the shape of these proteins, which allows it to kind of bring something in, like that flipase we just talked about. All right. Now, it is a barrier. It's not a perfect barrier. Things get through the membrane by accident all the time. All right. But generally, the factors that affect whether or not something can pass through the membrane are these four. You need to know these four. Size. So ions are tiny, but proteins are large. Polarity. All right. Charge and concentration. So know those four and know what they mean. So concentration, sometimes you need a lot of something before it can get in. Sometimes it's charge. You'll need to look up what it means by polarity. Is something polar? Is it look the same on both sides? So keep that in mind. These four, know those. High permeability. Well, gases, uncharged molecules get through pretty quick. Water is moderately permeable. Water can pass through just very slowly. Things are getting a little larger, and if they have some odd chemistry, like they're polar, then they have a harder time getting through. And then very low permeability is when you have something with a high charge, or it's very polar, or it's very large. All right, so just realize that some things get through, some things do not. Here is a list of those things. Cells maintain gradients. All right, an electrical gradient. I think we'll stop here. I'll start with the next one because this is a more complicated concept. But you can begin here looking through Chapter 5, and maybe this will help you see what's important and what is not.